Hello and welcome to another cardiovascular physiology video tutorial. I'm your host, Dr. James Colhane, and today we're going to be talking about an important endocrine system that's responsible for the regulation of blood pressure. I'm talking about, of course, the Raas system, the renin, angiotensin, aldosterone system. As I mentioned before, the Raas system plays an important role in the regulation of blood pressure, and it does so through two basic mechanisms: regulating systemic arterial vascular resistance, as well as controlling and changing kidney function, allowing the kidneys to hold on and retain sodium and water, thus increasing blood volume. Before we get into the overall blood pressure regulating effects of the Raas system, let's take a few minutes to discuss the biochemical nature of this particular system. The renin-angiotensin-aldosterone system essentially begins with a circulating enzyme that's produced by the liver called angiotensinogen. Angiotensinogen. Zoom in here for a second. Angiotensinogen, as I have here, is a protein approximately 400 amino acids in length. They're a little larger. This particular circulating protein is the first substance in a series of substances that will ultimately lead to the production of our active peptide angiotensin II. Let's see how this works here. Okay. All right. So as I pointed out, we start off with the peptide angiotensinogen. Angiotensinogen is converted to the next substance in this particular biochemical pathway, which is referred to as angiotensin I. How does this happen? Well, under certain circumstances, the kidney will release a substance, or an enzyme, from juxtaglomerular cells. These are specialized smooth muscle cells in afferent arterioles of the nephron. And will release a substance called, or an enzyme called, renin into the bloodstream. Renin is the enzyme responsible for cleaving off a portion of angiotensinogen. Actually, it's a 10 amino acid portion of angiotensinogen, leaving the rest of the molecule here. And that 10 amino acid sequence, or decapeptide, is referred to as angiotensin I. Now, when is renin released from the bloodstream? Well, or what are the signals for renin to be released into the bloodstream? There are a couple of things here. We have a decrease in blood volume is one signal, ultimately leading to a decrease in blood pressure. These are all signals here. We also have stimulation of the sympathetic nervous system will also stimulate renin release, and a decrease in delivery of sodium ions to the distal convoluted tubule, where the macula densa apparatus exists. So when any one of these events occur, and typically they occur in concert with one another, renin is released from the kidneys into the bloodstream, where it will begin to convert the protein angiotensinogen to the decapeptide angiotensin I. Okay. Now, angiotensin I will continue to circulate around in the bloodstream until it comes into contact with another enzyme that's located in various parts of the body, one of which is the lungs, and this enzyme is known as the angiotensin converting enzyme, or ACE for short. ACE is an interesting enzyme. It is located in blood vessels in the pulmonary tissues. There's also central nervous system ACE and ACE that is expressed on heart tissue. But when we talk about the Raas system, we tend to focus on the lungs as being the major source of ACE. ACE is an interesting enzyme because it is attached to the endothelial wall of blood vessels, small capillary blood vessels in the lungs. And as angiotensin 1 is circulating around in the blood, it will come through these vessels and actually be captured by or bind to the angiotensin converting enzyme. Once that binding happens, the enzyme gets turned on, cleaves off 2 amino acids, leaving us with the octopeptide angiotensin 2. So let's do that right here. Now, one of the questions you might ask is, why is this biochemical pathway important to know? Well, because the renin-angiotensin-aldosterone system plays such an important role in blood pressure regulation, a number of antihypertensive agents that we use to lower blood pressure actually interact with or interfere with this particular biochemical pathway. We have drugs that block the angiotensin converting enzyme, we have drugs that block renin, and we have drugs that block angiotensin 2's effect at the site of action, at its receptor. And so we're going to talk a little bit about what angiotensin 2 does next in order to actually increase blood pressure. Okay? All right. So let's talk a little bit about the vascular effects. Okay? So if we take a look up here, we have a blood vessel up here. Okay. And on these blood vessels, on arterial blood vessels, we have a receptor, a G protein coupled receptor referred to as the angiotensin 1 receptor. When angiotensin 2 is formed, when it's formed or produced in the bloodstream, it can circulate around and stimulate angiotensin 1 receptors. The result of this will be vasoconstriction. Now, typically, arteries are much more sensitive to the effects of angiotensin 2 than the veins are. So the vasoconstriction I'm talking about here is really arterial constriction. What this ends up doing is leading to an increase in systemic vascular resistance. And this SVR is essentially an indicator of how much resistance there is to blood flow in the cardiovascular system. And it's largely determined by how constricted or dilated your resistance arteries are in the body. Okay? Angiotensin 2 also has another effect. And this one is very important for kidney function. So we're going to go all the way over here to our kidney and to this little black dot on top of our kidney, which is our adrenal gland. Okay. Angiotensin 2 will stimulate AT1 receptors on the adrenal gland. And the adrenal gland, in response, will release a hormone called aldosterone. You get a blue marker here. Okay, so what we have here is we have the release of a hormone called aldosterone. This particular mineral corticoid-based receptor is very important in the regulation of kidney function. And in order to understand what it does to kidney function, okay, we have to go down here to my crude drawing of a nephron. If you have not studied nephron anatomy in a while, let's take a look at some of the different parts here. We have the Bowman's capsule. Okay, I'll just put a little BC here. We have the proximal convoluted tubule. Okay. We have the descending limb of the loop of Henle. We have the ascending limb of the loop of Henle. There's a thick and thin part. We have the distal convoluted tubule and the collecting ducts. Now, if you remember from your previous anatomy and physiology courses, The nephron is the individual functional unit of the kidney. This is where all the filtration and electrolyte regulation in the kidney occurs. Essentially what happens is blood is brought to each of the individual nephrons through tiny blood vessels called afferent arterioles. Once they reach the nephron, they form a tiny capillary bed inside the Bowman's capsule. As hydrostatic pressure and osmotic pressure change, water and electrolytes can get filtered out of the Bowman's capsule and into the lumen of the tubule. Anything that's left over exits the nephron via the efferent arteriole. And we have efferent. Okay? All right. Now, as I pointed out, water and electrolytes are forced through fenestrations in this capillary bed and enter into the tubular lumen. In the proximal convoluted tubule, there are a number of specialized protein transporters that will move electrolytes either out of the lumen or into the lumen from the bloodstream. One of the major electrolytes that is pumped out or moved out of the proximal convoluted tubule and into the bloodstream or reabsorbed is sodium. Okay. What's interesting about the PCT is that about 65% of filtered sodium is reabsorbed here. So a tremendous amount of sodium that gets reabsorbed through the Bowman's capsule and the glomerulus is immediately reabsorbed into the bloodstream. One of the things that is important to understand is that angiotensin II can actually stimulate this or enhance this. So when angiotensin II production occurs, we get even more sodium that's reabsorbed here in the proximal convoluted tubule. I won't get into nephron physiology in all that much detail because it's not really the point of this video tutorial, other than to say that this is one site of sodium reabsorption. The loop of Henle is another site of sodium reabsorption, as well as the distal convoluted tubule and collecting duct. Aldosterone, which remember is a substance that is released from the adrenal gland when angiotensin II is present. There are, remember, AT1 receptors here that are stimulated by angiotensin II. Aldosterone is released into the bloodstream. It's a mineral corticoid-based hormone. And it exerts its effect on renal function. Just going to come down through here. On the collecting duct. Okay, the last segment of the nephron that is able to reabsorb sodium and hold on to water. Okay, so let's take a look here at what happens in the collecting duct. Aldosterone actually stimulates the transcription and translation of specialized ion channels on epithelial cells here in the lumen that actually reabsorb sodium into the bloodstream. And the amount of these ion channels, or we call them the ENaCs, E-N-A-C, you'll find out in pharmacotherapeutics, the number of these ion channels that are present is directly related to aldosterone release. So aldosterone stimulates the production of these ion channels and their insertion into the lumen, and what we get is reabsorption of sodium. If you remember the old adage, where sodium goes, water will follow, and what we get here is a reabsorption of H2O. All of this will serve to increase blood volume. Okay? So all that will serve to increase blood volume. So if you remember, we started off our system here by saying that the Raas system, or we started off our video tutorial by saying the Raas system is an important system for the regulation of blood pressure. And again, it can elevate blood pressure or raise blood pressure by increasing systemic vascular resistance as well as increasing blood volume. Now, this video tutorial certainly doesn't get into all of the details of the Raas system. One of the things that I wanted to mention is that angiotensin converting enzyme, not only does it convert angiotensin 1 to angiotensin 2, but it also breaks down an important peptide substance called bradykinin. And bradykinin is a vasodilatory substance. So when Raas is activated, not only do we get a production of the vasoconstrictive angiotensin 2, but we also get the reduction in the amount of bradykinin in the bloodstream. One other thing that I wanted to also mention is that angiotensin 2 increases the release of antidiuretic hormone, okay, which can ultimately influence and work with aldosterone on this process down here. Okay? Again, understanding how the Raas system works is very, very important to understanding blood pressure regulation. And as I mentioned before, there are a number of therapeutic agents that we use to lower blood pressure that directly interact with this particular system. Well, that's all I have for today. I hope you enjoyed this video tutorial. Again, keep in mind that this isn't the entire system. This is a simplified version of Raas. However, I think it'll give you a good start in understanding in some of your studies on this particularly important endocrine system. This is your host, Dr. James Culhane, signing off. Good luck and have a great day.